What's your favorite organelle? Did you say mitochondria? Most people do. Most people say the mitochondria, the powerhouse of the cell, is my favorite organelle. Yes, it's true. The mitochondria does provide the power for our cells in the form of ATP, but it's actually, in my opinion, much more interesting for a few other reasons. The mitochondria actually has its own DNA, and that DNA that's in the mitochondria, these mitochondria were inherited not from dad and mom, but in most cases only from mom. How is that possible? Don't you inherit things equally from mom and dad? Well, in the case of mitochondria, that's not necessarily true. Mitochondria seem to only come from mom. So believe it or not, your powerhouse came from your mother. In this video, we'll explore a few other organelles, including the mitochondria, and get to the bottom of why our powerhouse is from mom. Let's get started. So in our second video here on A Tour of the Cell, we want to pick up where we left off from the previous video and start talking about the Golgi apparatus. The Golgi apparatus can be found here. It's often adjacent to the ER. And the Golgi apparatus basically is just some flattened membranes. Okay, so lots of membranes. And there's actually two sides. We got one side here, one side here. And depending on the orientation of these, it's hard to tell which one is which. So we actually define the sides by what they do. The cis side, which you can see I've labeled up here, is actually the receiving side. So notice here comes a vesicle, it's going to receive that vesicle, oh, here comes another. So this side up here, that's where we're receiving information. That's called the cis Golgi. The other side, that's where we're going to ship the vesicles away. So there goes one vesicle, there we go. That's the transport side or the shipping side. So you can probably, from this example, start to deduce what the Golgi does. If it's receiving here in the cis side and transporting here, basically it's kind of like a shipping and receiving department. Think of it as sort of like your local FedEx facility. So some packages come in, they're sorted, packaged, shipped, and then maybe transported somewhere else. We also know that in the Golgi, we have the correct folding of these new proteins that were made. And I'm going to go in more detail in a moment about that process of how we get these new proteins made. But the folding of those needs to happen very often there in the Golgi. The Golgi also produces lysosomes, which we'll learn about in a moment as well. So the Golgi has a cis and trans side, shipping, receiving, shipping, and it helps fold proteins and produce lysosomes. So, what about vacuoles? Sometimes vacuoles, you might also hear vesicles. A lot of times these words are used together. Basically, it's just a membrane-bound sac. And here's an example of one that you can see being made. So what happens, it's basically a little transfer ship, all right? So here's some product or some material that needs to be transported somewhere. So what happens is a little bit of the membrane pinches off, forming a little ship, a little vesicle, and then it comes out and it might bind with something else and then move on. So it's basically, if you think about it, just a little membrane-bound sac that is often used as for transfer of different materials. In this case, I've shown you a food vacuole. Right? So here's some food that's come in and it's going to bind with a lysosome, and I'll go over those more in detail in a moment, to start breaking down that food for use in the cell. So this particular one is a food vacuole. There are some other vacuoles. A contractile vacuole, as you can see here, we often find in freshwater protists, right? These small little organisms that live in freshwater. And because they live in freshwater, the water, because of osmosis, is often moving inside. So the water wants to come inside here. And the protist will often get too much. We don't want the protist to burst because of all the water flowing in. So it's evolved these contractile vacuoles, which contract and essentially shoot out the water that has moved in through the membrane. pumps out that excess water. So we've got the food vacuoles, we've got the contractile vacuoles, and then one that you see very often in plant cells is a central vacuole. So here's a nice plant cell, and what we see highlighted there is a central vacuole that is used to store. This helps reduce the overall surface area to volume ratio, or actually increase the surface area, because we can hold a lot of things in that vacuole, so therefore the volume doesn't seem quite as big in that cell. So I mentioned lysosomes already. Let's talk about what they are. Lysosomes are basically just these membrane-bound sacs that contain hydrolytic enzymes. So think of hydrolytic, right? We're going to break down something. So these enzymes very often are going to break down different particles or different food, perhaps. So here we go. What we've got here is typical lysosome. So it's got a membrane, and we've sort of like split it in half so you can see inside. Inside there, they look like little Pac-Mans. I guess that's our drawing for these enzymes. But you see these little enzymes inside? These enzymes are going to digest whatever they encounter. So the function, of course, is to digest these major macromolecules. So let's say we've got some food or some molecules that need to be digested. What happens is this lysosome will sort of hook up with this other membrane-bound vesicle. And what you'll see is that these little enzymes will move over, right? And they'll start to digest these larger molecules, break them apart. So if you think lys, right, and zome, a body, so this is just breaking down. Anytime you see lys, right? So it's breaking down, digesting these macromolecules. And this is important in the whole recycling program. So we need to recycle cell parts, break them down, and build them back up into what we need. And lysosomes also help us in apoptosis, which is programmed cell death. If a particular cell is no longer useful, or it's bad, or it's perhaps has a mutation in it that is not good, it's a damaged cell perhaps, and we need to break it down. We need the cell to die. So these lysosomes will actually help in that process. And then, of course, once that's been broken down, we can get rid of it if we need it, or we can use it for something else. So lysosomes breaking down macromolecules because they have these hydrolytic enzymes. One other interesting thing about lysosomes is that these are only found in animal cells. So you won't see these lysosomes in plant cells. So let's put some things together now. We call this the endomembrane system because this is all about all these membranes, all these organelles we've talked about that have membranes. So how this all works, putting it all together, let's say. Let's say there's some molecules that come into a cell. All right, so here's our cell, here's the outside, and a little bit of the membrane is going to pinch off and bring these molecules in. Okay, so it's formed a little vesicle or vacuole, a little transfer ship that's going to bring it in. Now what's going to happen with those? Well, I'm going to give you two scenarios. One, let's say that this is some food particles that needs to bind to a lysosome for digestion. Okay, so that's one route it can take. Another option is, let's say it's some sort of cell signal. Let's say that this cell has received a signal from another cell or another gland in the body. So that will come in, and what you'll see is we'll get this signal, this little molecule, and it will move to, the Golgi. Remember, the Golgi has a cis and a trans side. So we're going to receive some information here. It's going to sort it, package it, ship it, and then it's going to say, okay, now in this particular case, let's send it over, to the endoplasmic reticulum. Here, we're going to get a signal. Maybe that signal will go to the nucleus. Maybe it's a signal for something that we want to do. So let's just say that the signal that was received is one that, all right, DNA in the nucleus, we need the code. We need to make a particular protein. All right, so you remember the DNA, right, is held in the nucleus. So what's going to happen is it's going to make a copy of the gene we want, messenger RNA. It's going to send that out to these ribosomes. Now, do you remember what happens at ribosomes? Well, the ribosomes will make the protein that we need. So let's say we needed to make some insulin, right? You had a milkshake, you got a lot of sugar in your body, you need to break that down. So the nucleus is going to receive that signal, transcribe the DNA into RNA, right, the blueprint, send it out to the protein, say, all right, protein, send it out to the ribosome, make the protein, make us some insulin. Okay, so now that it's been made, we need to get it back out of the cell. So in the rough ER, there are those nice little ribosomes. A little bit of that will pinch off, as you can see here. Move back to the Golgi, sorted, packaged, shipped. Send it out now, maybe with some more. It's going to be now leaving in a secretory, right? It's going to be secreted vesicle and move out of the cell. Okay? So this is an example of how all of this works together, okay? Now, all of this is happening at the same time, right? Lots of things are going on in a cell. I've just given you a few little examples of some things that could occur, right? So just to review this one more time, because I think this can be tricky. If things come into a cell, right, they could have a few destinations. One might be to bind with a lysosome for degradation. One might be to go over to the Golgi, get packaged and shipped, head on over to the ER in the nucleus, send a signal. Then in the nucleus, right, we might have some transcription, send out to the ER, to those little ribosomes, say, hey, make this protein. This protein might be made, go back to the Golgi, sorted, packaged, shipped, then sends out of the cell for secretion. This is called the endomembrane system because the beautiful thing is that everything's just membrane, right? This is membrane, that's membrane, that's membrane, that's membrane, this is membrane, right? Nucleus inside is not. But then when it comes back out, it's all this membrane that's going to carry things out and into the cell. So we started talking about mitochondria. Let's move to mitochondria and chloroplasts as we start to wrap things up. So when you hear of mitochondria and chloroplasts, we often group these together because they are both similar and have some slight differences. So let's first talk about the similarities. So I've grouped this in a way that you can see the stuff here in the center is what's similar about the mitochondria and the chloroplasts. So what are some interesting things? One, they both have double membranes, right? You got one membrane, two membrane. One membrane, two membranes. Both have double membranes. They're both semi-autonomous, meaning self, right? They pretty much take care of themselves. They don't have to rely on a lot of other things to do their job. So they're semi-autonomous. They also have their own ribosomes. And you know ribosomes are what make protein. So they have their own. They don't have to rely on the ribosomes in the cell. They also, as I mentioned at the very beginning, have their own DNA, right? So they've got their own little DNA, and it happens to be in mitochondria here that you'll see this little circular DNA we saw early on. Both of them are involved in energy transformation because they both make ATP. Mitochondria make ATP from sugar. Chloroplasts, of course, make ATP, or our energy, from the sun. And the other thing you'll notice is lots of surface area. You see lots of membranes here, lots of membranes there. That's all about increasing surface area because that's where a lot of our reactions are going to take place. Now, how are they different? Mitochondria we find in both animal and plant cells. Wait a minute. I thought chloroplasts use the sun for their energy. Why do they need mitochondria? Well, there are times in plants' lives where the sun might not be available, and they might need some mitochondria so they can make energy for the cell. Mitochondria also break down sugar. Chloroplasts, of course, use the sun's energy. We find these in plant cells and photosynthetic algae. Okay, so mitochondria and chloroplasts have a lot of similarities, but as you can see, they have some differences as well. So let's get a little deeper into mitochondria specifically and go over the parts. So there's a typical mitochondria, and if we take it and sort of blow it up a little bit, there's these little structures inside the mitochondria. So basically, there's the double membrane, which we've already mentioned, double membrane, 1, 2. This would be the outer membrane, and then, of course, the inner membrane there. The inner membrane is highly folded. See all these folds? We call those cristae, okay? All those high level of folding. And, of course, that's about increasing the surface area. So now the intermembrane space is this fluid in between the outer membrane there and the inner membrane there. So this would be the intermembrane space. And when we learn about cellular respiration, we'll learn about that and what's really happening there. And then, of course, there's this fluid in the center. That's called the matrix. Also a great movie starring Keanu Reeves. So what does it do? Yes, powerhouse of the cell. But really, now that you're learning a little more about biology, really, mitochondria is all about the generation of ATP. ATP is our energy currency. This is what the cells use for energy. And that process that makes that ATP is cellular respiration. And we get that from breaking down sugar, fats, and some other fuels. Now, I started the whole video with this idea that your mitochondria, your powerhouse, is inherited from mom. Dad didn't contribute to your powerhouse. No. In fact, what we find is that the mitochondria are in the egg. There are a few studies which say the sperm has a very few mitochondria, but in most every single case, what we see is that your mitochondria you got from mom because the egg is really big. Remember, dad's sperm is just pretty small in comparison to this egg. So the mitochondria come with the egg and not the sperm. So since you get the egg from mom, sperm from dad, your mitochondria were inherited from mom, inherited from your mother. And that's almost always the case. All right, so chloroplasts. There it is. Chloroplasts we think of as being in plant cells. What do? Well, photosynthesis, of course. What's their structure? Well, like mitochondria, we see an outer and an inner membrane, right? So here's our outer membrane that we're showing you. Here's the inner membrane inside. And then, of course, the space in between the outer and the membrane, outer and inner membrane is the intermembrane space. Inside the chloroplasts are these other flattened sacs, and these are just membranes as well. One of those is called a thylakoid. So one of these membranes is a thylakoid. There's a thylakoid. There's a thylakoid there. There's a thylakoid membrane. A stack of thylakoid membranes is called a granum. Okay, that's a stack of those membranes. The fluid is called the stroma. Now don't confuse this with the stomata, the opening in leaves, but the fluid in the chloroplast is called stroma, internal fluid-filled space. What do? Photosynthesis, right? They are essentially taking the sun's energy and converting it into a chemical form of energy for the plant cell. Okay, so we see a lot of the same similarities that we saw in mitochondria, inner, outer membrane, this space, lots of membranes. Why? Why is there so many membranes? Surface area. These reactions are taking place on these membranes. That is needed for all that surface area. Okay, so how did mitochondria and chloroplasts evolve, right? They seem to be kind of independent, kind of advanced. How did we get these mitochondria and chloroplasts in us? Well, there is a theory about how mitochondria and chloroplasts originated, how they evolved. And the idea is that some ancient bacteria were engulfed by an ancestor eukaryotic cell. So think of animals and plants, right? We're eukaryotic. We have eukaryotic cells. So at one point, the idea is that, all right, these cells were hanging out, right? Some of the membranes started to come inside and form some organelles, right? Some of the outer membrane started to come inside, started to form little other organelles. And eventually, one of these ancient bacteria, which just happened to process sugar and make energy was engulfed. All right, so this life form ate that life form. Well, over time, they started a symbiotic relationship, meaning that this ancient bacteria started to live symbiotically inside this other organism. Well, over time, right, we think those evolved into our modern day mitochondria, and then the same thing would have happened with the chloroplast, right? Some photosynthetic ancient bacteria was engulfed, and it became modern day chloroplasts. So you may say, well, what's the evidence of that, right? Well, one, they're semi-autonomous. I told you that to begin with, right? The chloroplast and the mitochondria kind of act like they're their own things from the beginning anyway. Well, and that makes sense if they were originally their own. They also have their own circular chromosomes. And we know that bacteria, their chromosomes are circular, right? So they have their own DNA, they kind of do their own thing. So that's why this theory is really pretty credible, because we think they were engulfed because they were actually ancient bacteria engulfed by these ancestral eukaryotes. Okay, so hopefully now after watching these two videos, you can feel comfortable labeling these cells. So what I'm going to do is I'm going to say in just a moment to pause the video, see if you can actually go around and label each organelle in the cell, and then I'll come and help you a little more. I'm going to get you started with a few that I didn't mention in either video. These are what you might consider less obvious, right? All of these are important, but these are maybe a few that we kind of omitted. The plasma membrane down here, we're going to have a whole podcast on that, a whole video. So I didn't mention it in this video because we'll have a whole one dedicated to that later. Right here, we see a cytoskeleton. This is just helping, right? Think of the skeleton of your body, just provide structure and support. We also have a centrosome. These are used in mitosis, in motility, right? And then we also have a peroxisome. Peroxisomes are basically places where these little oxidation reactions in the body can occur. And we also get some lipid synthesis from those. Okay, so these are the ones I hadn't mentioned. I think you can do the rest on your own. So pause me for a moment, see if you can label them on your own, and then I'll help. Okay, how did you do? All right, did you get them all? So this is the animal cell, right? Animal cell. So here we've got a vacuole, large vacuole there. These are lysosomes, right? You see the digestive enzymes. Mitochondria, of course, we just learned about those. Moving over, oh, here's the Golgi. Cis and trans side might be hard to see in this example unless we know which one's coming and going. Then, of course, we have the smooth ER because there's no ribosomes. Here's the ribosomes. There they are. Those are free-floating ribosomes. But notice this ER has ribosomes, so that's rough ER. These last two, right, we've obviously got this nucleus. Do you remember what that's called? The place in the center, that's the nucleolus. Okay, so think about how you did on those. It is important to be able to identify them by shape, by structure, but I think more importantly is, did you know what each of those did or what they do in the cell? Okay, so I've got one more little thing for you. Now, see if you can label this plant cell. So pause me for a moment. Try to go around and label all of those. Okay, so let's look here. How'd you do? Same thing. I'm going to tell you, right? There's the membrane, plasma membrane. This one may have got you there. A cytoskeleton. Hopefully you remembered that from the last one. And peroxisome. But what about this one? That's a cell wall. We haven't talked about that much, but plant cells have the additional cell wall. You may remember that's made of cellulose, that rigid polysaccharide. Okay, then you probably got the Golgi. You probably got the ER, the smooth ER, and the rough ER, and the ribosomes again. You got the nucleus, large central vacuole here in the plant cell. Hopefully you got mitochondria. Don't forget, mitochondria are in plant cells. And then also what's unique to the plant cell is the chloroplast. So that concludes 2 videos on all the organelles in the cell. Hopefully this helped in your understanding and comprehension of all of these parts of a cell and how they work to make your cell function.